Today we are going to discuss conjugated lipids. And before going to discuss the topic, I will repeat the definition of lipids. Lipids are esters of fatty acid with alcohols, and they are non-polar hydrophobic compounds which are soluble only in organic solvents. Now, as far as the classification of lipids is concerned, lipids are classified into three classes: simple lipids, conjugated lipids, and derived lipids. You have been through the simple lipids, which includes neutral fats. The example of neutral fats is triglycerides and waxes. Then we will go for the conjugated lipids, which are further subclassified as phospholipids, glycolipids, sulfolipids, and lipoproteins. Then the third category is that of the derived lipids. So derived lipids includes, derived lipids are those compounds which are obtained from the breakdown of the lipids. They are fatty acids, cholesterol, and other steroid molecules. And it also includes the fat soluble vitamins like vitamin A, E, and K. We have been through this classification of lipids they were classified into three major classes: simple, conjugated, and derived. And it is you can see conjugated lipids includes phospholipids, galactolipids, and lipoprotein. Now, conjugated lipids are also called as compound lipids. And in compound lipids, as we know, fatty acids are esters of lipids are esters of fatty acid and alcohol. So in addition to fatty acids and alcohols, these conjugated lipids also contain some other compounds like phosphorus, amino nitrogen, or carbohydrates, etc. That's why they are called as conjugated. Or we can define compound lipids as esters of fatty acid containing group in addition to an alcohol and a fatty acid. The first class of this conjugated lipids is phospholipids, as the name is suggesting that beside fatty acids and alcohol, which is glycerol, they contain phosphate group. So as you know that glycerol is a 3-carbon structure. So two fatty acids will attach to the glycerol molecule, and the third carbon of the glycerol molecule will attach to the phosphorus compound. Now, as I told you earlier, this is the diagrammatic presentation of a phospholipids, and remember, phospholipids are amphipathic molecules. And we know that glycerol is a 3-carbon structure. So at carbon number 1 and 2, the two fatty acids are attached. And you can see in this picture, this is a saturated fatty acid which is attached at carbon number 1. At carbon number 2, we have the unsaturated fatty acids, and this kink is showing that this is a saturated fatty acid with a double bond. And at the carbon number 3, we have the phosphate group which is attached. So being highly negatively charged molecule, this phosphate makes this phospholipid as an amphipathic molecule. Amphipathic molecules are those molecules which have the characteristics of both hydrophobic and hydrophilic qualities. So phospholipids, this is the basic structure of a phospholipid, and they are amphipathic molecules. In the previous slide, we just discussed the basic structure of the phospholipids. Phospholipids or glycerophospholipids, these are synonyms. And as we came to know that glycerol being a 3-carbon structure, and it has two fatty acids attached at position carbon number 1 and carbon number 2 by means of an ester linkage. So in lipids we have ester linkage. Remember, in carbohydrates we have glycosidic linkage, and in proteins we have the peptide linkage. And at carbon number 3, the phosphate group is attached. Now this phosphate groups in different phospholipids can again be attached to various alcohols by means of an ester linkage. Remember, alcohols have got OH group in their structure, and that group which can be attached to the phosphate, it can be choline. When a choline attached to a phospholipid, then that phospholipid is called phosphatidylcholine or lecithin. This group can be ethanolamine and it can be inositol. So choline, ethanolamine and inositol have hydroxyl group in their structure. So that hydroxyl group of these compounds can again be attached to the phosphate group by means of an ester linkage, and on the basis of these phospholipids are being labeled as phosphatidylcholine, phosphatidylethanolamine or phosphatidylinositol. We have one more important phospholipid that is called as cardiolipin, and in the next slide we will discuss the structure of cardiolipin and its importance. But remember one thing, this cardiolipin, a phospholipid, it is a component of the inner mitochondrial membrane, and we all know very well that this inner mitochondrial membrane has a role in the generation of ATP from the fuel. Now this is the basic structure of a phospholipid. You can see this is the first fatty acid attached to the carbon number 1. This is the second fatty acid attached to the carbon number 2 of the glycerol molecule, and carbon number 3 of the glycerol molecule has got phosphate group attached to it, and this X indicate the different groups which can be attached by means of the ester linkage to this phosphate group, as we discussed in the previous slide. And this X can be a choline. So this is the chemical formula for choline, and remember one thing, you are not supposed to memorize these formulas. You just remember the name, that it can be a choline, this is it can be an ethanolamine, it can be a serine molecule, or it can be an inositol molecule. Or we can see here that one more this is a structure of the X group is the glycerol. So this these are the various groups with their chemical formulas which can be attached to the phosphate molecule by means of the ester linkage. So these are the chemical formulas, and you are not supposed to memorize these formulas. This is the diagram of a phosphatidylcholine, and you can see here, as I told you, that these phospholipids are amphipathic molecules. So this part, which is comprised of the phosphate and the choline, it makes it the hydrophilic part, and this hydrophilic part is associated with the water, and the hydrocarbon chain of this fatty acid is comprised is hydrophobic in nature, so it is being buried into the interior of the cell. And here we can see that on carbon number 1 usually the saturated fatty acid is attached, where on the carbon number 2 we have the unsaturated fatty acid, and here we can see here that this unsaturated fatty acid has a double bond which is of cis configuration. If you recall the cis and the trans isomerism, so in cis isomerism the functional groups are on the same side of the this hydrocarbon chain, and you know that these cis cis double bond produce a kink in the structure of the phospholipid, and we know, being a component of the lipid bilayer, this kinking has play a very important role in maintaining the fluidity of the cell membrane. If you recall the fluid mosaic model of the plasma membrane, that this unsaturated fatty acids with the cis double bond produce a kink, and because of this the fluidity of the cell membrane is being maintained. This table shows you the different types of conjugated lipids which are present in the plasma membrane or the cell membrane of the human beings or mammalian cell. So as far as this phospholipids are concerned, we have just been through this, that it can be phosphatidylcholine, and its another name is lecithin. It can be phosphatidylethanolamine with the name cephalin, phosphatidylserine, phosphatidylthreonine, phosphatidylglycerol, and phosphatidylinositol. If you recall, while discussing this lipid bilayer of the plasma membrane, we discussed these different phospholipids in detail, and also we will discuss cardiolipin in the next few slides. Beside those phospholipids, other conjugated lipids which are the part of the lipid bilayer of the cell membrane, they include sphingolipids, that is sphingomyelin, and glycolipids. Glycolipids means when the lipids conjugate with the carbohydrates, and these glycolipids can be cerebrosides, sulfatidides, and gangliosides. And beside these conjugated lipids, we have steroid molecule in the form of cholesterol as a part of the plasma membrane. This is a structure of phospholipid that is called as a cardiolipin. This cardiolipin is also called diphosphatidylglycerol. You can see that in the center we have a glycerol molecule, and to this glycerol molecule two phosphatidic acid moieties are attached. So you can see the 2-phosphatidic acid attached to a glycerol molecule in the center. So it means it forms a dimer, and it has the two highly negatively charged phosphate group attached to this. This is the structure of cardiolipin, and cardiolipin are exclusively found in the inner mitochondrial membrane, as I discussed in the previous slides, and that it has a role mainly with the production of the ATP from the reduced coenzymes or from the reducing equivalents, and also it has a role, it interacts with the electron carriers in the electron transport chain. We have been through the structure of the cardiolipin, and as I told you that it is a dimeric phospholipid that exists exclusively in the inner mitochondrial membrane of the eukaryotic cell, and it has two chiral carbons, and if you recall, it has 4 fatty acids attached uh, to the glycerol molecule, and it is having the 2 phosphate groups which make it a highly negatively charged molecule, and this enables this phospholipid to interact with the electron carriers in the respiratory chain, and those are the substrate which are present in the mitochondria for ATP production. Here we will, in this slide, we will recall the function of the phospholipids which we already covered while going through the structure of cell and the plasma membrane. And we are very well aware that these phospholipids are the basic constituents of the lipid bilayer. And since they have an amphipathic property, so they form double layered membrane with the water soluble molecules on the outside of the cell membrane and the water insoluble molecules inside of the cell. Some of these phospholipids act as an activator of enzyme, and if you recall, hormones are first messengers. There are many hormones which when reaches their target tissue, they cannot enter into their target cells, so they mediate their action via second messenger. So one of the phospholipid, which is the phosphatidylinositol, act as a second messenger, and it plays a role in the transmission of hormonal signals to the interior of the cell. Then we have phosphatidylcholine, which act as a lung surfactant. It decreases the surface tension, and it helps in the expansion of the lungs in a newborn. Deficiency of this phosphatidylcholine can lead to the respiratory distress syndrome, especially in premature neonates. Now, we have already discussed the role of phosphatidylcholine, that phosphatidylcholine act as a surfactant, and surfactants are the compound which decreases the surface tension in the alveoli of the lungs, and they play a role in the expansion of lungs at the time of the birth in a newborn. So when there is a deficiency of phosphatidylcholine, it can lead to the respiratory distress syndrome in the newborn and especially in the premature babies. Here you can see there that in the presence of the phosphatidylcholine in normal concentration, we can see the alveoli, they are fully expansion of the alveoli, and in the absence of this phosphatidylcholine, you can see the collapsed alveoli. Here I will must share with you an important this aspect of this phosphatidylcholine, that body in the brain utilizes this phosphatidylcholine for the synthesis of a neurotransmitter that is called as acetylcholine, and acetylcholine has a role in regards to the memory, retention of memory. Now here we will discuss the second type of the lipids which are normally present in the lipid bilayer, and these are called the sphingolipids. These sphingolipids are mainly present in the nerve cells and the brain tissue. And if you recall the definition of a lipid, that they are the esters of fatty acid with alcohol. So in phospholipids, the alcohol was glycerol, but in sphingolipids, they don't contain glycerol, rather they contain an amino alcohol which is called as sphingosine. This sphingosine and a fatty acid, when they are being attached, they form a structure that is called ceramide. So ceramide is the basic structure of the sphingolipid, and this when this sphingolipid, a phosphate group with the choline is attached to this, then it is called sphingomyelin. So sphingolipids can be on the basis of the alcohol group. In sphingolipids, the alcohol group is an amino alcohol which is called as sphingosine. It can be a sphingophospholipid, or they are simply sphingolipids. Sphingomyelin is a sphingophospholipid. It contains a a phosphocholine molecule attached to it, and other sphingolipids are called cerebrosides and gangliosides. So we will discuss these one by one. This is the structure of a sphingophospholipid that is called sphingomyelin. Now this red structure is a structure of sphingosine. Sphingosine is an amino alcohol, and to this amino alcohol, when a fatty acid is attached, then it is called ceramide. So ceramide is the combination of sphingosine with the fatty acid. When to this structure a phosphate group along with the choline is attached, then it is called sphingomyelin. And you are very well aware that this sphingomyelin is mainly present in the nerve cell. It's present in the myelin sheath which surrounds the axons of the nerve cells. Now this sphingomyelin is very important in regards to being a component of the myelin sheath. Now what happens that there is an enzyme which is called sphingomyelinase. When this enzyme is deficient, then the degradation of this sphingomyelin cannot take place in the lysosome, and it can lead to a disorder that is called as a Niemann-Pick's disease. Niemann-Pick's disease is a lysosomal storage disease, and as a result of this, there is an accumulation of sphingomyelin in the lysosomes. Now, glucocerebroside. This is a structure of glucocerebroside. Glucocerebroside is mainly comprised of this sugar, sphingosine, the red structure, and when this sphingosine combines with the fatty acid, it forms ceramide. So ceramide is the basic structure, and when to this ceramide a carbohydrate, and if it is a monosaccharide, glucose, then it is called glucocerebroside. So glucocerebroside is mainly made up of ceramide plus glucose, and you can see here this is the glucose which is attached to this uh, uh, fatty acid and the this sphingosine alcohol. In the previous slide, we just discussed sphingolipids, and sphingolipids were those lipids where the alcohol is sphingosine. And sphingolipids can be sphingophospholipid, that is sphingomyelin, or they can be cerebrocytes or gangliosides. When we say cerebrocytes or ganglioside, then to this sphingosine along with the fatty acid, carbohydrates are attached. These carbohydrates can be in the form of monomeric residues, or they may be in the form of oligosaccharide. So this is simply the structure of a glycolipid. So cerebrocytes and ganglioside, they are glycolipids. Only the difference is that in case of cerebrocytes, only a single sugar residue is attached to the lipid part, where in the ganglioside it is an oligosaccharide chain. So this is a simple version of the glycolipid. You can see that these are the lipid molecules, and to this the oligosaccharide chain is attached, and it is the part of the lipid bilayer of the cell membrane. Glycolipids can be subclassified as cerebrocytes and gangliosides. You have to understand the difference between cerebrocytes and gangliosides. In cerebrocyte, as we saw, that the basic structure is ceramide. What is ceramide? It is combination of sphingosine with a fatty acid. This is the basic structure. Now, when to this structure a monosaccharide is attached, either it is glucose or it is galactose, then it is called cerebrocytes. When the glucose molecule is attached to the ceramide, it is called glucocerebroside, or when galactose is attached to this ceramide structure, it is called galactocerebrosides. So cerebrosides are ceramide plus monosaccharide. When we say and glucocerebrosides have a specific function, especially in the cell membrane of the macrophages, whereas the galactocerebrosides, they are found exclusively in the membranes of the brain cells. So they have a special function especially in the brain cells. Now you are very well aware that cerebrosides are ceramide plus monosaccharide. This monosaccharide can be glucose or it can be galactose. Gangliosides, they have carbohydrates in the form of oligosaccharide chain. And this oligosaccharide chain may contain salic acid residues in their structure. Salic acid or N-acetyl-neuraminic acid, it is a monosaccharide derivative, and in the oligosaccharide chain there may be monosaccharides or monosaccharide derivatives may be there, and they are also called gangliosides because they were first identified in the ganglion cells of the central nervous system. So now you are very well aware that in gangliosides the number of monosaccharide residues are more than 2, and they are in the form of oligosaccharide chain, and this oligosaccharide chain may contain monosaccharides or monosaccharide derivatives, and especially salic acid, which is called NANA or N-acetyl-neuraminic acid. Now the nomenclature which is being used to identify this gangliosides is based on two things. Number one is that the number of salic acid residues present in the structure and the sequence of the carbohydrate. We call them GM. GM means ganglioside having a single salic acid residues in its structure. M stands for mono. So similarly, GD means when the 2 salic acid residues, GT means when the 3, and GQ means when the 4 salic acid residues are present in the ganglioside structure respectively. As we were discussing regarding the nomenclature of the gangliosides, we discussed that GM means when the ganglioside having a single the salic acid residue in its structure, And when with this GM number 1 is added, means GM1, it means that it is on the basis of their mobility on the chromatograph. So GM1 gangliosides, they migrate slowly on chromatograph, thin layer chromatograph. So we have three types of ganglioside: GM1, GM2, and GM3. So on the basis of the mobility of these gangliosides on chromatograph, they are again being classified as GM1, GM2, and GM3. So the number of the salic acid residues present in the ganglioside structure and the sequence of the carbohydrates and their mobility. These are the three criteria for as far as the nomenclature of the ganglioside is concerned. Now this oligosaccharide chain which is attached on the ganglioside, it serves a very important function, and it is being used as a surface marker, and it has a role in the cellular recognition and cell to cell interaction. So this is the only carbohydrate part which is playing a role, because these gangliosides, cerebrosides, they are glycolipids, and if you recall, they are present on the plasma membrane, on the outer side of the plasma membrane. So they have a special role in cell to cell recognition, as well as they have a role in the determination of this blood group antigens. Here is the structure of GM1. It means ganglioside having one salic acid residue in its structure, and which moves slowly on the chromatographic field. Now this is the basic structure, that is, it is lactosyl ceramide. As I told you, the basic structure is made up of ceramide, and ceramide is a combination of sphingosine alcohol with a fatty acid, and it is called lactosyl, means a lactose molecule. You know, glucose and galactose, they form lactose. So this is the basic structure, lactosyl ceramide, and you can see to this an acetyl galactosamine, a glucose residue, and this is salic acid. So only one salic acid is present in the structure, oligosaccharide chain of the GM1 ganglioside, and uh, one indicates its mobility on the chromatographic field. You all are very well aware with the apoptosis. Apoptosis is a programmed cell death, so that when a old cell is replaced with the new one, so the old cell undergo degradation. And every cell has got a lysosome which is responsible for the intracellular digestion. So deficiency of lysosomal enzyme, which are mainly concerned with the breakdown of the cerebrosides and the gangliosides, when they are deficient, when we say deficiency of enzyme, mainly it is because of the genetic defects or mutation in the gene which code for that particular enzyme. So lysosomal disorders are genetic disorders. Genetic disorders are those disorders where there is a mutation in the gene which code for that particular enzyme. So there are two disorders which are mainly, there are many disorders. We will just discuss two of these. One is which is related to the degradation of the ganglioside, that is called as Tay-Sachs disease. And in Tay-Sachs disease, the enzyme which is mainly concerned with the breakdown of the ganglioside, it is deficient. The Jewish children are mainly affected by this disorder, and it results in the death in early years of the life. Gaucher's disease actually is a disorder which is related to the degradation of the glucocerebroside. The glucosidase enzyme is deficient, and what happens, the glucocerebroside, they get accumulated in the lysosomes of various organs like liver, spleen, and it result in the enlargement of the liver and spleen and severe anemia. So these are the two genetic disorders which are concerned with the degradation of the gangliosides and the cerebrosides. Another class of the conjugated lipids is lipoproteins. As the name suggests, that when protein conjugates with the lipid part, then it forms lipoproteins. And the major purpose of this lipoprotein formation is the transportation of lipids in the plasma or in the circulation. You are very well aware that only water soluble components can be transported across plasma or in the blood. So lipoproteins are actually complex particles which are comprised of two components: a central hydrophobic core, which is comprised of cholesterol esters and triglycerides. Now here, cholesterol esters are actually when a cholesterol molecule attached to a fatty acid, then it's called esterified cholesterol or cholesterol ester. And you are all very well aware with the TAG, triacylglycerides or triacylglycerol. So the central hydrophobic core is comprised of cholesterol esters and TAG, and this central hydrophobic core is then covered by a hydrophilic membrane, which is mainly composed of phospholipids, free cholesterol, and apoproteins. Apoproteins are the protein part which conjugate with the lipid molecules, and these apoproteins are designated as apoprotein A, B, C, and E. This is a schematic presentation of a lipoprotein molecule. You can see this is central hydrophobic core, and this is esterified cholesterol molecule, and this fork-like structure are the TAGs. So the central hydrophobic core is comprised of the cholesterol esters and TAG. What are cholesterol esters? When a cholesterol molecule combines with a fatty acid by means of an ester linkage. Now this central hydrophobic core is then covered by a hydrophilic membrane, and that membrane is comprised of phospholipids. These are the apoproteins, you can see, and this is the free cholesterol. So this hydrophilic membrane part is mainly composed of these proteins, apoproteins, cholesterol, and you know that free cholesterol is again an amphipathic molecule because of the hydroxyl group at the carbon number 3, and these apoproteins which make it lipoproteins as the water soluble so that they can be transported in the circulation. When we say lipoproteins, it means when the lipids conjugate with the protein, and proteins are having a density of more than 1.3 grams per ml, and this, on the basis of this protein part, this lipoproteins are classified into chylomicrons, very low density lipoproteins, low density lipoproteins, that is called as the LDL molecule, and HDL, high density lipoproteins. So the density of the lipoproteins depends upon the ratio of lipids to proteins. When we say high density lipoproteins, it means the protein content is high and the lipid part is less. So chylomicrons, they have more lipid content and they have a small amount of protein as apoproteins, than very low density. So density is mainly concerned with the protein content. As the density increases, the amount of protein in the lipoprotein molecule increases. So we have four major classes of the lipoproteins. As far as the function of lipoprotein is concerned, I told you earlier that lipid has to conjugate with proteins for transportation. So chylomicrons, CM stands for chylomicrons, and chylomicrons, they transfer when we consume dietary lipids. After digestion and absorption, that dietary lipids from intestine is transported to the peripheral tissues and mainly to the liver by in the form of chylomicrons. So chylomicrons are responsible for the transportation of dietary lipids from intestine to the peripheral tissues. VLDL and LDL, the very low density lipoprotein and low density lipoproteins, they transfer endogenous lipids. Endogenous lipids are those lipids which are being synthesized in the liver and they are being packaged there in the form of VLDL. So the VLDL starts its journey from the liver, and in the peripheral tissues, it while going through the plasma and the circulation, it there converts into LDL molecules. So VLDL and LDL are mainly responsible for the transportation of endogenously synthesized, when means those lipids which are synthesized within the body in the liver, and they are being transported to the peripheral tissues for utilization. HDL, high density lipoproteins, remember they have a high protein content and less lipids, and they are responsible for transportation of cholesterol from peripheral tissues towards the liver for their final metabolism. So there is a reverse cholesterol transport. Mainly HDL carries cholesterol from peripheral tissues to the liver for final disposal, and in the HDL the cholesterol is mainly transported in the form of esterified cholesterol, when the cholesterol is esterified with a fatty acid. Now you are now very well aware that the purpose of formation of lipoprotein is transportation of lipids into the circulation within the body from to various cells and tissues. So here this chart shows you the various types of lipoproteins and the lipid content they transport. You can see chylomicrons has less cholesterol, but it has the high content of TAG, and as we discussed earlier that it transport the dietary TAGs and cholesterol from GIT to the peripheral tissues for utilization. Very low density lipoproteins, they are synthesized mainly in the liver. They are being packaged there in the liver, and they do contain a small amount of cholesterol, and they have a high content of TAG. Now this TAG is being transported from liver to the peripheral tissues for utilization. Now if you recall enzymes, that we have certain plasma enzymes, and one of these plasma enzymes which are present in the quite sufficient amount is the lipoprotein lipase enzyme. Lipoprotein lipase, the substrate for lipoprotein lipase is triacylglycerol. So you can see here that the chylomicrons and VLDL, they have a high content of TAG, and these lipoprotein lipase, they are present in on the endothelial lining of the capillaries. So when the TAG is being circulated in the plasma, they target the TAG and they convert TAG into free fatty acid and glycerol for the cellular utilization. So when the chylomicron starts its journey from GIT towards the peripheral tissue, so what happens? Its TAG is being acted by the lipoprotein lipase. Its TAG content is decreased. You can see here the content of TAG is high, and in chylomicron remnants the content of TAG is decreased, because when it was being transported from GIT to the peripheral tissues, the lipoprotein lipase has acted on TAG, and the remnant is being then taken up by the liver. And then we have ideal intermediate density lipoproteins. Here we can see now the cholesterol content is gradually increasing, and as well as ideal has a TAG component. And then we have the LDL. LDL mainly is responsible for the transportation of cholesterol, because when the VLDL started its journey from the liver and is going to the peripheral tissues, the TAG was hydrolyzed by this lipoprotein lipase. So TAG content is decreased, but the cholesterol amount is there, and this is the reason why the LDL molecule is called bad cholesterol. So LDL mainly transports cholesterol from the liver to the peripheral tissue, as well as it is responsible for the transportation of fat soluble vitamins. HDL also carries a considerable amount of cholesterol, but this cholesterol is not the free cholesterol, but it is a esterified cholesterol, and we know that this is a reverse transportation from peripheral tissues to the liver for their final disposal or for metabolism. And HDL has in some extent as an antioxidant role. Uh, this slide not only shows that the functions these various lipoproteins perform in our body, But along with that, their clinical significance is also given, that when there's a deficiency of lipoprotein lipase. If you recall, chylomicrons, they carry the dietary TAG. So they have a huge amount of dietary TAG, and that dietary TAG has been broken down by the lipoprotein lipase. So what happens if there is a deficiency of lipoprotein lipase? The amount of chylomicron in the circulation is increased, and it gives milky appearance to the serum when it is being taken for investigation. And as far as the VLDL is concerned, we are very well aware that VLDL is synthesized in the liver. It carries the endogenous triacylglycerol, and it carries that TAG for utilization by the peripheral tissues. So, defects in the VLDL synthesis in the liver, because liver is the site for the endogenous TAG synthesis. So, if there is a defect in the their synthesis, so it will lead to fatty liver. Again, we are very well aware that LDL is responsible for the transportation of endogenously synthesized cholesterol from liver to the peripheral tissue. Due to any reason, there are many reasons which can lead to increased concentration of LDL into the circulation. And if the LDL concentration is increased in the circulation, it can lead to atherosclerosis, that is the narrowing of the lumen of the vessels. And similar is the case that if there is a decreased concentration of HDL, HDL performs a very good function. It carries cholesterol from peripheral tissue to the liver for their final disposal. So if there is something, if the levels of the HDL are less, again it is a sign, it is a marker that this person may suffer from atherosclerosis. So high LDL and low HDL, both are markers of atherosclerosis, that is the narrowing of the blood vessels. Here we will briefly discuss about the protein part of the lipoprotein, which is called apoproteins. And apoproteins, actually, when conjugate with the lipid part, they make this lipid to be transported into the circulation. So they transport lipids in blood, in CSF and lymph. We are very well aware that the lipid components of the lipoproteins, they are water insoluble. Now these proteins, they play a key role in normal lipid homeostasis by transporting cholesterol and lipids between cells. These apoproteins, they are mainly synthesized in the liver and the intestine, but a small amount of ApoB. If you recall, these apoproteins can be designated as ApoA, B, C, and E. So ApoB can be made in the heart tissues and ApoE by the macrophages. All tissues in the body, they are capable of degrading these apoproteins, but mainly they are degraded in the liver. This chart shows us the different type of apoproteins, and ApoB100. It is a structural component of the VLD. It is a structural component of the VLDL, IDL, and LDL. And the major thing regarding this ApoB100 is that it is a structural component of the VLDL, IDL, and LDL. And we know that the lipoprotein VLDL starts its journey from the liver, and while going through the plasma, it converts into IDL and then into the low density lipoprotein. So then when this low density lipoprotein reaches to the peripheral tissue, then this LDL particle is taken up by that particular cell or tissue by means of receptor mediated uptake. And receptor only recognizes LDL particle because of the presence of the ApoB100. Similarly, ApoB48, it is a part of the chylomicron remnant, because what is chylomicron remnant? Chylomicron remnant is that the dietary lipids, when they start their journey, they are in the form of chylomicron. While going through circulation, the TAG of that chylomicron is targeted by the lipoprotein lipase. So its TAG content is decreased. So when it reaches to the liver, it is in the very small amount. That's why it is called as chylomicron remnant. And liver takes up this chylomicron remnant by recognizing this ApoB48. So these apoproteins have a role in the recognition of the receptor mediated uptake of these various lipoproteins. And there's a very interesting fact regarding these apoproteins, that these lipoproteins, they exchange their apoproteins in the plasma. ApoE mainly is a part of this chylomicron, VLDL, IDL, and HDL, and as I told you that these various lipoproteins, they exchange their apoproteins, and this ApoE actually has a role in the binding of the IDL and remnants to the LDL. So ApoA1, it is a structural component of HDL and chylomicron. And mainly what happens that as I told you that HDL, high density lipoprotein, has a role in the reverse transportation from peripheral tissues to the liver, and it carries cholesterol in the esterified form. So for esterification of cholesterol, what is an esterified cholesterol? That when a cholesterol combines with the fatty acid, for that we need an enzyme LCAT. So LCAT is required for the formation of esterified cholesterols, and the HDL carries esterified cholesterol from peripheral tissues to the liver. So this ApoA1, not only a structural component of the HDL and the chylomicron, but it is activator of the enzyme lecithin cholesterol acyl transferase. LCAT stands for lecithin cholesterol acyl transferase enzyme. Now this apoprotein A2 is again a structural component of HDL and chylomicron, but its function so far is not known. ApoC1 is activator of LCAT. If you recall, ApoA1 was also an activator of LCAT. And ApoC2 is an activator of lipoprotein lipase. Now you can see here that chylomicrons, VLDL, IDL, they all have a high cholesterol content, TAG content. So lipoprotein lipase, the substrate for this enzyme is TAG. So ApoC2 is an activator of lipoprotein lipase. ApoC1 and ApoA1, they are activators of LCAT enzyme. And ApoC3 is an inhibitor of lipoprotein lipase enzyme. So one thing is very clear regarding these apoproteins, that these apoproteins, they make this lipid insoluble particles into a transportable form. Number two, they are activators of enzymes. And number three, they have a role in the receptor mediated uptake of these LDL in the peripheral tissues.